Hey people, get ready to crush everyday TOEFL iBT tests for all modules and make your dreams come true. I'm your motivator and I'll be guiding you through the test prep videos. So don't forget to like, share, and subscribe or you'll be missing out on some serious preparation.
Conversation 1. You will hear a conversation. A female student visits the University Health Center to make an appointment. First, you have some time to look at the 5 questions. Now listen carefully and answer the questions. Hi, I was hoping to schedule an appointment with a doctor. I've been having these really bad headaches for the past week or so.
I'm sorry to hear that. Let me pull up the schedule. Have you been seen at the health center before?
Yeah, I came in last semester for a flu shot, but that's about it. I'm not sure if my records are still in the system.
Don't worry, once you're registered, your file stays active for the duration of your enrollment. Okay, so I see your information here. Now, can you tell me a little more about the headaches? That way I can direct you to the right practitioner.
Well, they started about a week ago. At first I thought it was just stress, finals are coming up, but they've been getting worse. Sometimes the pain is so bad I can't even look at my laptop screen.
That does sound concerning. Have you had any changes in vision, nausea, anything like that?
Actually, yeah, I've noticed some blurry vision, especially later in the day. No nausea though.
Okay. Given the vision issues, I think we should get you in with Dr. Patel. She's our general practitioner, but she also has experience with neurological referrals in case that becomes necessary. Now, here's the thing, her earliest opening isn't until Thursday afternoon. But if the symptoms get significantly worse before then, I want you to come to our urgent walk-in clinic. It's open every weekday morning from 8 to 11.
Thursday, that's 3 days from now. I was really hoping to get in sooner. Is there any chance of a cancellation or something?
I understand your frustration. Tell you what, I'll put you on the cancellation list. If someone cancels before Thursday, we'll call you right away. In the meantime, are you taking anything for the pain?
Just over-the-counter ibuprofen. It helps a little, but it wears off pretty quickly.
That's fine for now, but try not to exceed the recommended dosage. And I'd strongly suggest you cut back on screen time where you can. I know that's tough with finals, but it might help with both the headaches and the blurry vision.
Yeah, easier said than done, right? But I'll try. Should I bring anything to the appointment?
Just your student ID. And if you wear glasses or contacts, bring your current prescription information if you have it. Dr. Patel may want to do a basic vision screening as part of the exam.
Okay, great. Thanks so much for your help. I really appreciate it.
Of course. We'll see you Thursday at 2:30, if not sooner. Take care of yourself.
That is the end of the conversation 1. You now have some time to answer the questions. Conversation 2. You will hear a conversation. A male student visits the university computer lab to get technical support. First, you have some time to look at the 5 questions. Now listen carefully and answer the questions.
Excuse me, are you the tech support person on duty? I'm having a problem with one of the lab computers.
Yes, I am. I'm Miss Chen, one of the IT support specialists. What seems to be the issue?
Well, I was working on my senior thesis. It's a pretty large document with a lot of images and charts, and I needed to save it to my cloud drive through the university portal. But every time I try to upload it, I get an error message saying the connection timed out.
Okay. Is this happening on a specific workstation, or have you tried others?
I've only tried the one I was sitting at station 14, over by the window. I didn't want to lose my work by switching computers.
That's understandable. You won't lose anything if you save locally first, though. But let me check something. We actually had a network patch installed last night, and a few of the machines on the east side of the lab, which is right where station 14 is, have been having intermittent connectivity issues since then. So it's probably not anything you did.
Oh, that's a relief. I thought maybe my file was corrupted or something. It's due to my advisor by Friday, so I was starting to panic a little.
No, your file should be perfectly fine. Here's what I'd recommend. For now, save a copy to a USB drive as a backup. Do you have one with you?
No, I don't actually. I usually just rely on the cloud storage.
No problem. We keep a small stock of USB drives at the front desk. You can borrow one and return it when you're done. They're 8 gigabytes, which should be more than enough. Meanwhile, I'm going to move you to station 22 on the west side. The network connection over there has been stable all morning.
Okay, that sounds good. But, I also had another question. I was trying to use the statistical software, DataLab Pro, to generate some of my charts, and it kept freezing every time I tried to export a high-resolution image.
Yes. That's actually a known issue with the current version we have installed. The newer version, 4.2, fixes that bug. We're planning to roll out the update across all lab machines during the maintenance window this weekend. In the meantime, there's a workaround. If you export as a standard resolution first and then use the image editor, we have Photoscope installed on all machines. You can upscale it without losing too much quality.
Oh, I didn't know Photoscope was available. That could actually work. Is it hard to use?
Not at all. It's pretty intuitive. And if you get stuck, we have a quick start guide on the IT support website under the software tutorials section. Just search for Photoscope.
Great. You've been really helpful. One last thing, is the lab going to be open late this week? I might need to come back and put in some extra hours before my deadline.
Good timing on that question. Starting this week, because it's the final weeks of the semester, we're extending our hours. The lab will be open until midnight Sunday through Thursday, instead of the usual 10 p.m. closing. Fridays and Saturdays stay the same, we close at 8.
That's perfect. Thanks so much, Ms. Chen. I'll grab that USB drive and head over to station 22.
Sounds like a plan. And don't hesitate to come back if you run into any other issues. Good luck with your thesis. That is the end of the conversation too. You now have some time to answer the questions. Lecture 1. You will hear part of a lecture about anthropology. Tool use in primates. First, you have some time to look at the 6 questions. Now listen carefully and answer the questions.
Today we're going to look at one of the most fascinating areas in anthropology, tool use among non-human primates. For a long time, tool use was considered a uniquely human trait, something that separated us from the rest of the animal kingdom. But starting with Jane Goodall's groundbreaking observations in 1960, we've had to completely rethink that assumption. Goodall observed chimpanzees in Ngam, Tanzania, stripping leaves off twigs and inserting those modified twigs into termite mounds to extract termites. Now, this was revolutionary because the chimps weren't just using objects, they were actually modifying objects for a specific purpose. That's tool manufacture, not merely tool use, and it forced scientists to either redefine what it means to be human or redefine what we mean by tools. Since then, researchers have documented a wide range of tool use behaviors across primate species. Capuchin monkeys in Brazil, for instance, use stones as hammers and anvils to crack open nuts. Orangutans in Borneo have been seen using sticks to extract seeds from hard-shelled fruit. And in West Africa, chimpanzee populations have developed what we might call cultural traditions, specific tool use techniques that are passed down from one generation to the next through social learning.
Student, Professor, is there evidence that different chimpanzee groups use different tools for the same task? Like regional differences?
Excellent question. Yes, absolutely. This is actually one of the strongest arguments for primate culture. Researchers like Andrew Whiten and Christoph Bosch have catalogued dozens of behaviors that vary between chimpanzee communities but cannot be explained by ecological or genetic differences. One group might use stones to crack nuts while a neighboring group, with access to the same stones and the same nuts, never does. The only plausible explanation is social transmission, which is essentially what we call culture. Now, I want to draw an important distinction here between what we see in primates and what we see in, say, crows or sea otters, which also use tools. In most non-primate species, tool use appears to be largely innate or individually learned. But in primates, especially great apes, there's strong evidence of cumulative cultural evolution, where techniques are refined and built upon over generations. This doesn't reach the complexity we see in human technology, of course, but it suggests that the cognitive foundations for technological development have deep evolutionary roots.
That is the end of the lecture 1. You now have some time to answer the questions. Lecture 2. You will hear part of a lecture about physics. Wave-particle duality. First, you have some time to look at the 6 questions. Now listen carefully and answer the questions.
Alright, so last time we wrapped up our unit on classical optics, reflection, refraction, interference, all phenomena that are beautifully explained by treating light as a wave. Today, though, we're going to see how that wave picture, as successful as it is, only tells half the story. We're diving into wave-particle duality, one of the central concepts of quantum mechanics. Let's start with the problem that launched the quantum revolution, blackbody radiation. In the late 1800s, physicists tried to predict the spectrum of radiation emitted by a heated object using classical wave theory, and the result was catastrophic, literally called the ultraviolet catastrophe because the theory predicted infinite energy output at short wavelengths. In 1900, Max Planck resolved this by proposing that energy is emitted not continuously, but in discrete packets he called quanta. Each quantum carries energy proportional to its frequency, E equals h times f, where h is Planck's constant. Then in 1905, Einstein took Planck's idea further. He proposed that light itself is quantized into particles, which we now call photons. He used this to explain the photoelectric effect, the observation that shining light on a metal surface ejects electrons, but only if the light's frequency is above a certain threshold. Classical wave theory couldn't explain why intensity didn't matter below that threshold, but Einstein's photon model could. Each photon delivers a discrete packet of energy to a single electron, and if that packet isn't large enough, no electron is ejected regardless of how many photons hit the surface.
Student, but Professor Chen, if light is made of particles, how do we explain interference patterns? Doesn't that require waves?
That is exactly the right question, and it's precisely the tension at the heart of wave-particle duality. The answer is, light genuinely behaves as both. The double-slit experiment demonstrates this beautifully. When you send photons one at a time through two narrow slits, each photon hits the detector at a single point, that's particle behavior. But if you accumulate thousands of these individual hits, they form an interference pattern, that's wave behavior. Somehow, each single photon seems to interfere with itself, as if it passes through both slits simultaneously. And here's where it gets even stranger. In 1924, Louis de Broglie proposed that this duality isn't limited to light. He suggested that all matter has wave-like properties, with a wavelength inversely proportional to its momentum. This was confirmed experimentally in 1927 when Davison and Germer showed that electrons, which everyone agreed were particles, produced diffraction patterns when bounced off a nickel crystal. So electrons behave like waves too. The implication is profound: wave-particle duality is not a quirk of light. It is a fundamental feature of nature at the quantum scale.
That is the end of the lecture too. You now have some time to answer the questions. Lecture 3. You will hear part of a lecture about medicine. Immune system response. First, you have some time to look at the 6 questions. Now listen carefully and answer the questions.
Today I want to walk you through the basics of how the human immune system responds to a pathogen, say, a virus that enters through the respiratory tract. The immune response is typically divided into 2 major branches: the innate immune response and the adaptive immune response. They work together, but they operate on very different timescales and with very different levels of specificity. The innate immune system is your first line of defense, and it kicks in almost immediately, within minutes to hours of infection. It includes physical barriers like the skin and mucous membranes, but also cellular components like macrophages and neutrophils. These are phagocytic cells, meaning they engulf and destroy pathogens. They recognize broad molecular patterns shared by many microorganisms, what we call pathogen-associated molecular patterns, or PAMPs, through receptors called toll-like receptors. The key point is that the innate response is fast but nonspecific. It doesn't distinguish between one virus and another, it just recognizes that something foreign is present. Now, while the innate system is holding the line, it's also activating the adaptive immune system, and this is where things get really sophisticated. Dendritic cells, which are part of the innate system, capture fragments of the pathogen, called antigens, and migrate to the lymph nodes. There, they present these antigens to T cells. This is called antigen presentation, and it's the critical bridge between innate and adaptive immunity.
Student, Professor Okafor, what happens after the T cells recognize the antigen? How do they know which pathogen to target?
Great question. So each T cell carries a unique receptor on its surface that recognizes one specific antigen shape. When a dendritic cell presents an antigen that matches a particular T cell's receptor, that T cell becomes activated. It then proliferates rapidly, clonal expansion, we call it, producing thousands of copies of itself. Some of these become cytotoxic T cells, or killer T cells, which hunt down and destroy infected cells directly. Others become helper T cells, which coordinate the broader immune response, including activating B cells. B cells are equally important. Once activated by helper T cells, B cells differentiate into plasma cells that produce antibodies, proteins specifically shaped to bind to the pathogen's antigens. These antibodies circulate in the blood and neutralize the pathogen by blocking its ability to infect new cells, or by tagging it for destruction by other immune cells. The whole adaptive response takes about 7 to 10 days to fully ramp up during a first exposure, which is why you feel sick for several days before recovering. But here's the truly remarkable part: after the infection is cleared, a subset of both T cells and B cells persist as memory cells. These memory cells can survive for years, even decades. If the same pathogen enters the body again, the memory cells mount a response that is far faster and far stronger than the original, often neutralizing the threat before you even develop symptoms. This is the principle behind vaccination: you introduce a harmless form of the antigen to generate memory cells without causing disease.
That is the end of the lecture 3. You now have some time to answer the questions. That is the end of the TOEFL iBT listening test. You now have some time to review your answers.
Hey people, how was your test? Don't forget to like, share, and subscribe, or you'll be missing out on some serious fun. See you tomorrow!